some diseases can inhibit the amino acid import from the lumen across the apical membrane. So let's have a look at them. Well, they're autosomal recessive diseases, first of all, Hartnups and cystinuria. And what happens is that these are defects in apical amino acid import. But for some reason, people who have Hartnup and cystinuria, they don't have protein deficiency. And this is evidence for multiple transport systems, which we can see here, the B0 plus system, the B0 system, the EAAT for anionic amino acids, and the aminoglycine proline cotransporter. That's the fact that they don't have protein deficiency, despite this apical defect, suggests that there's multiple systems. It also suggests that there's a separate oligopeptide transport system, which, as I talked about, is PEP-T1, hydrogen cotransport. So you can still absorb oligopeptides, so therefore these aren't too serious in terms of protein deficiency. So first of all, looking at Hartnup's disease, well, what does that do? Well, Hartnup's disease inhibits B0 cotransporter, which transports neutral amino acids along with sodium across the apical membrane of the enterocytes into the enterocytes, right? So therefore, less neutral amino acids are absorbed, but oligopeptides are still absorbed because you have PEP-T1, right? And the signs, so because of the neutral amino acids not being absorbed, the signs, you'll get dermatitis and brain ataxia, so lack of coordination. Now, cystinuria, on the other hand, what that does is it inhibits the B0 plus cationic and cysteine neutral amino acid antiporter, right? So you're going to get a lack of cationic amino acids absorbed. But also, this transporter, this antiporter, is also on the convoluted tube of the kidneys, and it allows the reabsorption of cysteine from the urine back into the blood. So if you have cystinuria, what happens is that less cysteine is reabsorbed from the urine into the blood, and as a result you'll have high cysteine in the urine, which will result in kidney stones. So what I described so far is the method by which proteins are digested by luminal digestion and then membrane digestion. They're absorbed from the apical membrane, and then they're effluxed from the basolateral membrane. But some proteins can be absorbed directly via two routes, and the purpose of this is an immune response. So let's have a look at that. Well, the apical proteins / polypeptides, they can either enter the enterocytes by phagocytosis, in which case 90% of them are degraded by lysosomes and about 10% are absorbed into the body. Or they can be phagocytosed by M cells in Peyer's patches. Well, what are Peyer's patches? Peyer's patches are the patches of lymphoid tissue in the gut. And what M cells do is they phagocytose these proteins. Then they package them in vesicles which are coated in clathrin, and they secrete these vesicles basolaterally into the lamina propria, which is the tissue on the basal side of the enterocytes. And then what happens is that immune cells, they essentially transfer the proteins to the immune cells. So the immune cells, they process the antigens of the protein, transfer them to lymphocytes for an immune response. So you can probably guess that these proteins that are coming in, they're harmful. They could be as a result of bacteria or viruses. They're being phagocytosed by M cells in Peyer's patches, packaged, secreted, and the immune cells process them for the antigens to be transferred to lymphocytes for an immune response. So essentially, there's protein uptake for mucosal immunity. And as I mentioned at the start of the protein section, is that not only for they take proteins taken up intact in an intact form for an immune response by M cells, but infants, when they drink breast milk colostrum, they absorb intact proteins through endocytosis.